Good afternoon, everybody, and welcome to our distinguished seminar today. I'm absolutely elated to introduce our distinguished speaker for today, Professor David Meaney from the University of Pennsylvania. He is the chair of the Department of Bioengineering at UPenn, and I know that he's been there as a faculty member for 20 years. Prior to that, he got his Ph.D. and his master's at Penn, and before that, he got his bachelor's in biomedical engineering at RPI, Rensselaer Polytechnic Institute. He now holds the Pollack Endowed Chair in engineering, and he also holds a joint appointment with neurosurgery because his work relates to the central nervous system. He studies repair mechanisms, he studies growth, studies regeneration, and obviously those areas have a lot of applicability in neurosurgery. His work is very well respected in the field. He has published papers in very prestigious journals, and his list of honors and awards is also quite significant. He has received the STAB award, the Wasifong Investigator Award. He has received all kinds of other recognitions of his research. I have known Professor Meaney for a number of years. I think maybe the first time that we met, we were both participating in an injury center at the University of Alabama at Birmingham, if I'm not mistaken. And I was fascinated by the work then, and I continue to be fascinated today. So I really look forward to this presentation. So before I turn it over to him, I wanted to indicate that yesterday the U.S. News came out with its rankings, and my dear friend here, as chair of the department, I'm sure he's very happy to note that UPenn Bioengineering went from number 10 to number 6. So it's a great recognition for the Penn Bioengineering program. So let us welcome Professor Dave Meaney to UC Davis.
So Barry, thank you for that introduction. And what I'll ask throughout my talk is if my voice starts to drop a little bit and I can't be heard in the back, if somebody could just give me one of these signals, that would be great. It's certainly a pleasure to be here. I appreciate the invitation. Davis has an impressive program that has great, what I would call dynamic range. And you have investigators at different length scales of the problems in biomedicine, and you also have a great range of the types of investigations that are going on here. And I have to say that I'm quite envious of some of the labs that you have. The team lab that you have here is quite impressive. I have my own ideas that I'm taking back with me, and I'm going to be pounding on my dean's door to replicate some of these things. So what I wanted to talk about today was some of the work that we've been doing for the past few years now, and it's developing into I think an area of research, not just for my lab, but for some of the collaborators that I've been working with, which really focuses on this aspect of studying the neurological disease at multi-length scales. The brain has one of these unique properties of having complexity at almost every scale. And with that, and studying some of this, for some reason my slides are not advancing. I guess my talk is very quick. So let me just try this one more time. So one of the challenges with the brain is that you have this complex structure at many length scales, some of which is illustrated right here. My career actually started looking at brain as a very soft material, describing the structure of the soft material, but moving quickly into more cellular and molecular neuroscience questions. And really the intriguing part of the system of the brain, at least for us now, is going down to the synaptic structure. And when we look at the number of synapses that form these delicate connections among all the neurons in the circuitry within the brain, one of the challenges, not just of our lab, but in fact many, if not all neuroscience labs, is how to take events that occur at this length scale, at the individual synapse level even, and move it up to understanding how it influences behavior of circuits in important brain regions. This is the hippocampus of the brain, and how it reflects in disease processes. And as my talk indicated, the title of my talk, we've been studying this whole disease of traumatic brain injury for some time. Early part of my career was focusing on how to prevent this injury and subsequently the disease from happening in the first place. And more recently, we've been looking at the origin of the changes that occur after an injury or a concussive injury, and how that may regulate some of the changes in the circuitry. So I always have this slide up here, and I mentioned it at lunch today with the students because this traumatic brain injury, or in earlier forms, they simply called it traumatic head injury, has this classic injury pyramid, where if you look at the relative incidence of the injury based on its severity, the tip of the iceberg are these severe cases. So if you look at statistics from the Centers for Disease Control, approximately 30,000 people each year fall into this category. Moderate injuries, which are defined clinically as injuries where you can see something on CT, or the person hits a certain threshold of neurologic deficits, there's about another 150,000 of these. And the remarkable thing about this pyramid, of course, is that as you get towards the base, it builds out. You have over 1 million people a year in the mild category. So this pyramid grows out quite a bit when you think about the incidence of this injury in population, in the US population. These are statistics just for the US. If you look at the statistics in the emerging world, because as more and more people in the emerging world are driving automobiles, the incidence rate and the rate at which this is happening is increasing exponentially. And of course, in the public press, we've heard a lot recently about the effects, the long-term effects of these types of injuries. And that's really, for the field, how we've moved from considering it an acute episode, where we focus really on trying to help promote recovery from that acute episode. And understanding now, even that acute episode, no matter what we try to do, may lead to a disease state. And often what we hear about in people returning from the war is this high incidence, for instance, of post-traumatic stress disorder, and how that's related to the IED blast exposures that they experience while on patrol. We've known for a while now in boxers, the studies in boxers, and in fact the definition of dementia pugilistica was originated in boxers several decades ago, and how these brains of boxers who's experienced a number of concussions over their career will look like the brains of much older people because they show these neurodegenerative changes in their 40s and not in their 80s. And when we consider this, and we look at the field in general, and I'm going to narrow in soon on what we've been studying, there are a lot of studies in the field that initially tried to characterize this injury in terms of its biochemistry. The other aspect that often was a common feature in these studies was if we could only understand how neurons die or degenerate within certain regions of the brain, we'd be much better off in terms of promoting recovery strategies. And then finally, more recently, there's been an awareness that even in situations which mild injuries fall under, even in situations where you don't lose many, if any, neurons within the brain, you have a change in the synaptic connections. So this is a classic multi-scale problem, because if you especially consider some of the changes in the synaptic connections, the strength, the weights of these connections over time, and how it influences the microcircuit behavior, eventually, and we're now seeing in clinical studies, changes in the widespread networks of the brain, or the so-called default mode networks, which are ultimately manifest in some of the impairments we see in these populations. So this is really an outline of what I'm going to talk about today. I'm going to present some highlights of the work that we published. I'm also going to give you some insights into some of the more recent work that's now under consideration. And what I'm going to do is I'm going to focus mainly on these two areas where we in the lab are building in vitro systems that are models of these concussive injuries. And because they're in vitro, we have a much more precise control over what we expose these circuits to. And by doing that in tandem with in silico models of literally the circuit activity and the conductivity of the circuit, we can go back and forth in a design sense on understanding how alterations of what we see experimentally can influence patterns created in these networks, and in fact we measure patterns of these networks through some high-speed calcium imaging. And in the long term, we can periodically take this information and spin it out into in vivo therapies. I'm not going to talk at all about in vivo therapies. We've had some recently that have been generated from this work. Maybe in my closing comments I can talk about it. And really, the length scale in this talk is moving from identifying what I would call mechanosensors that are localized at the synapse. And of all those that are possible candidates, we've identified one, and how that mechanosensor changes its physiology, and that change in the physiology can immediately affect the microcircuits, and ultimately the plasticity, or the ability of these circuits to learn new patterns, encode new patterns after the traumatic event. And as a general outline, we're ultimately driven by the challenge of restoring this activity of the network to regain function, and I'll comment on that at the end. Okay, so I thought I would first talk about some of the unique features of this injury as it progresses to a disease. So I'm going to show you a couple of video clips. The first video clip, for all of you that are NFL fans, you may have seen this because often what happens nowadays when you see a concussion on the field is that it's replayed several hundred times on ESPN, on all the shows, and it's almost burned into your memory. And the reason why I showed this clip is for the simple reason that if you look at this play and understand what's happening in this play, it really is one example of just how complex and unique this injury is. So if you look at this over time, this is played back in real time. You can see these two players here that are now on the ground. This is a situation where both the striking player and the struck player were concussed. And now it's shown in slow motion. And so the receiver, you can see as you look at it and you do kind of a biomechanics recreation in your mind, he's really not struck directly in the head. He's actually struck more towards the bottom. And I put that in there because that's the Patriots' Wes Welker. I'm a Bills fan. I'm a Patriots fan, a Patriots player. But if we go back and let me just replay the tape because I want to come back to this play again. Two players were concussed, a striking player and struck player. Mechanically, these are different events for each player. What happens if you track the head motions that occur in each of these players? They're unique and distinct from one another. And in fact, if you take this and look at the entire clinical population that has a concussion, every person has a unique mechanical etiology to their concussion. And what we now know, and what's been part of some of the regulations that are coming on board in the National Highway Traffic Safety Administration, is we now have models, finite element-based models, that we can use to literally provide a window into the brain to understand mechanically what's happening in the brain during these kinds of events. This is a video clip which shows you the earliest version of these finite element models. This is now becoming part of the safety testing standard for any car that's sold in the United States in 2 years. What you have to do is you have to crash the car into a rigid fixed barrier. There's a test dummy that is an instrumented test dummy that will move forward, and the airbag you'll see deploys in this test. There's an acceleration that the head experiences. And what they now do is they take these accelerations and they compute some magical number which tells the tester if this car design passes or not in this test. The government has now expanded this considerably and has used this acceleration input to derive a computational model. And it's this computational model that is the first window into the brain itself and how the brain moves and deforms within the skull. So again, this is an early model. This is a very simple model. It's kind of an ellipsoidal shape. It has some structure to it. Some of the more recent models actually have much more anatomic detail to it. And if you look at this situation and you try to estimate what parts of the brain experience the kinds of deformations and the nature of these deformations, you really end up having what I would call a design criteria for simulating these rapid, complex events now ultimately in a culture dish. So what we've done, it's really not rocket science at all, but we take a very traditional type of cell testing system that many are familiar with. This goes back a couple of decades with the flex cell system. This green grid here is a transparent, optically transparent silicone membrane, and cells are plated on this entire surface. These are primary cortical neurons that you see here. They form these connections, and this entire system, shown in an expanded view here, can be mounted on a microscope. And by pressurizing the top surface, one of the things that happens when you pressurize this top surface is that you can deflect the membrane downward. And obviously, if you can deflect the membrane downward with a fixed pressure and increase the pressure, you can slowly increase the strain that's applied to the membrane. Since the cells are attached to the membrane, they in turn stretch themselves. So all of this happens very quickly in our system. This is an event when you estimate what happens in the brain. This deformation occurs in about 25 milliseconds. It's faster than you can snap your fingers. And the entire event, the impact event itself, is typically over and done with within 100 milliseconds. So this is something that takes some timing. We've used the simplest approach, which is to dynamically deform and return these neurons to their pre-stretched state. And we've used this over and over again, not just in primary cortical neurons, but also in cell lines that are expressing recombinant receptors. And that's what I'm going to talk about in the next section, because our question, when we start to recreate the mechanical conditions associated with concussion, is to zone in on the synaptic structure, and in particular the postsynaptic surface, and ask which of these receptors that are commonly expressed at the postsynaptic surface, which of these are responding, if you will, are sensing this mechanical signal. So this is a part of a talk where I present one cartoon slide which represents about 2.5 years of a graduate student's work. Not to belittle the graduate student in any way. This is a tremendous amount of work, because what we did is we took a model system, PAC293 cells, and we expressed these recombinant receptors and all the different variations that you can see from NMDA receptors, AMPA receptors, metabotropic glutamate receptors, even a little bit of voltage-gated calcium channels. And we systematically scanned the entire population of important postsynaptic glutamate receptors at the excitatory synapses. This shows some of the complexity that we used in this approach, because these glutamate receptors are formed from distinct subunits, and depending on the type of subunits that they're assembled from, so for instance, AMPA receptors, they can create what they call homomeric AMPA receptors, which are GluA1 homomers. They have distinct properties when they're activated. There are other kinds of receptors. We focused a lot on the NMDA receptors, which have a certain expression subtype based on the subunit that can change developmentally in mature or immature circuits, and also are localized either within the synaptic cleft, postsynaptic surface, or outside in the extrasynaptic space. So we systematically scan these. The nice thing about this system is we can co-transfect with a fluorescent marker to identify ones that are successfully transfected. And long story short, what we found from all of that is the single receptor that was responding most to these mechanical forces that mimicked concussion was the NMDA receptor. So the NMDA receptor is a common glutamate receptor that people study in learning and memory. It's also a receptor that is often a focus of study when we study disease and diseases like ischemia and excitotoxicity, because the receptor can be overactivated, and by overactivating the receptor in these disease states, you can cause neuronal degeneration. This was the first glimpse that all of the work that we were doing in the HEC293 cells would translate to primary cortical neurons. So what we see here is the result of an experiment where our response is measured with a calcium-sensitive fluorescence dye. So the ability of, or the function, one of the functions of the NMDA receptor is to flux calcium, as well as a couple of other ions. And because you can use a calcium indicator to indicate when it's activated, it's a nice readout of when you activate these receptors. So in primary cortical neurons, when you stretch the receptor, you get this nice normalized response, which you can block if you antagonize only the synaptic NMDA receptors, and you can block completely when you antagonize or block both synaptic and extrasynaptic receptors. And we're going to come back to this kind of work time and time again, and we're going to go forward into HEC293 cultures and back into the primary cortical cultures to examine some of the changes in the circuitry. So with this evidence that it's the NMDA receptor is the primary receptor, one of the things that we then explored was to dive into the structure of the receptor. So just because there's a mixed audience here, I just wanted to review some of the important properties of the receptor, because we're going to start manipulating some of these properties and talk about them later. First, it's a receptor that has a coagonist of glutamate and glycine that are binding to it. The reason why it's called NMDA receptor is because an alternative agonist is NMDA itself that has a selectivity for it. One of the interesting properties of this receptor is that it's formed by distinct subunits. There's always the GLU-N1 subunit, and it's partnered with GLU-N2, GLU-N3 subunits of different types. I also made it a point here to look at some of the complexity of the intracellular domain of the receptor, because it complexes with a lot of structures and proteins. And for our work, interestingly, it also complexes with cytoskeleton, which gives us an idea that there's a physical association with the cytoskeleton that gives rise to this activation. And as I mentioned before, it is one of the primary sources of calcium influx from the glutamate receptors. And importantly, the physiology of this receptor at resting membrane potentials, which on this graph is right about here. If you look when you activate the receptor with its coagonist, the amount of current that's fluxed through the receptor is somewhat limited because you have this natural magnesium block of the receptor. So in normal 2 millimolar magnesium, you really don't flux much because magnesium sits in this pore-forming region right here. And one of the things that happens, and you see dramatically in the electrophysiology, is when you bathe it in 0 millimolar magnesium, there's no magnesium to limit the flux through the pore here, the ion channel, and you get a significant increase in this flux. So it's a magnesium block, which is a common feature of NMDA receptor of different subtypes, and it's something I'll come back to. And then finally, I'll just call your attention to some of the organizational features of the receptor. I'm going to go through and I'm going to talk about how this receptor is formed from different subunits, the NR1, GLU-N2A, and GLU-N2B. This is primarily a synaptic composition, and an alternative form is GLU-N1 and GLU-N2A, and extrasynaptic receptors are largely GLU-N1 and GLU-N2B, although there's some debate about that in the literature. So this goes back to now looking at the HEC293 system, because the beauty of this system, of course, is that you can express any combination of these receptor subtypes that you want. You have a nice readout with a calcium indicator dye. This is something where we systematically scan all the different subtypes that are commonly found in the cortex and in the hippocampus. These are two important regions in traumatic brain injury which are often damaged, and we wanted to see if there's any unique mechanosensing properties. And indeed what we found, and what you see just summarized quickly here, is that the sensitivity of the receptor to this mechanical perturbation is really due to the GLU-N2B subunit. So that when you express a receptor, recombinant receptor that has the GLU-N2B subunit, you get this nice increase in response. And when you don't have it and you only have GLUN2A plus GLUN1, you get no response at all. So that gave us an indication that there was some specificity to this. This is just a bar graph showing you the same data that I went through here. But we then systematically looked at the regulation and at the different subtypes that are found in both the cortex and the hippocampus, and including a more complex or triheteromeric form, which includes the NR1, NR2B, and NR2A. And the intriguing part of this story is that if you compare the response after a mechanical stretch, you find that, as I mentioned earlier, when you look at the NR1, NR2A, you get very little response. And there's an intermediate response of the triheteromeric formulation that is now fully exposed, if you will, with the GLUN1, GLUN2B. And this is in stark contrast to the kind of response that you see with these recombinant receptors when you just add NMDA. So there's no chemical diversity, chemical agonist diversity, like there is a mechanical agonist diversity. And it's one feature that when we look forward, we think that it might distinguish chemical toxicity in the brain from this mechanical toxicity. And I'll comment on that in a few slides. We then went back to some earlier publications. This was from a science paper from Les Satton's group and Earl Ellis at MCV. And they were the first to show that if you patch on using whole cell patch clamp and you look at uninjured cells, you get this nice J-shaped curve, which indicates the physiology of the NMDA receptor as a classic magnesium block. What's intriguing about this observation was that when you stretched these cells using the similar kind of system to what we've been using in our lab, you immediately see a loss in the magnesium block. So what you're doing is you're altering the physiology of the receptor. At the time, there wasn't an ability to go and then systematically scan for subtype specificity. The same graduate student that did the HEC293 work now looked in primary cortical neurons and found that indeed in uninjured neurons you get this nice J-shaped curve, and in neurons where you block all of the NMDA receptors except for the receptors containing this GLUN2B subunit, when you examine the stretch response, you find that this GLUN2B subunit containing receptors are the ones that are showing this loss in the magnesium block. So we've now provided mechanistically, based on some earlier evidence in the literature, a reason at the physiological level of what's happening to this receptor immediately following this injury. Now, there's a collaborator at Children's Hospital of Philadelphia, and for those of you that know Penn sits right next to CHOP, Children's Hospital of Philadelphia. It's a great environment because you can literally walk over in about 5 minutes' time and find a number of collaborators that can add quite a bit to the story. And one of the collaborators in the work I'm going to talk about now is with Dave Lynch at the Department of Neurology at CHOP. And Dave has studied NMDA receptors for some time. And the field has identified many of the domains, the important domains of this receptor and the different subunits. The glutamate binding domain for the GLUN2B, some of the transmembrane domains, the pore-forming domains for the receptor, intracellular domains. And what we did is we looked at first the domain, in fact only exclusively looked at the domain within the cell itself, the intracellular domain, which spans these amino acids shown here. And Dave had a number of truncated mutants that we could express in the HEC293 system. You could express, and we did, express a truncated mutant which eliminated some of the binding at the extreme end of the subunit to a couple of important synaptic signaling proteins. There was some evidence in the literature that this indirectly coupled to the cytoskeleton. We expressed this, and we saw no change in mechanosensitivity. So this domain, this region right here, was not responsible for this mechanosensing ability. We started to selectively chop it up. Of course, as engineers, we thought about the optimal way of doing it, which was rather than sequentially going through it, we jumped all the way and cleaved off the whole thing. And Dave kind of shuddered a little bit, and he said, look, in biology, you actually systematically go through these things rather than going to the shortest path. To this day, I don't regret it, because what we found economically, that when you clip off the entire domain associated with coupling to the cytoskeleton, we eliminated the entire response. So the sensitivity was somewhere on this domain. And to make a long story short, we found that there were regions where we could tune down this mechanosensitivity. Not eliminate it, but we can tune it down. And then Dave had some mutants that he constructed in his lab, because within those domains there were important phosphorylation sites that regulated the function of the receptor based on the literature. And we systematically expressed each of these. And what's shown here is a result of all of this. This is from Pollack in my lab, who's now graduated. And this blinking red light right here, this bar, identifies the domain on the GluN2B subunit that can take the stretch response, which again is normalized here, and reduces it by about 60%. So we don't tune it down completely, but this one amino acid in the entire subunit can really dramatically change this ability of the receptor to sense. We have not looked at combinations of these phosphorylation sites, but what it has led us to, which we're working on right now, is since this site is the target of a couple of kinases, it brings the idea forward that phosphorylation and the regulation of phosphorylation can indirectly tune this mechanosensitivity. And there was some indirect evidence from some studies a few years ago in our lab that we could tune this phosphorylation, but at the time we didn't know mechanistically what was going on. So at this point we realized that we had an evolving story of what we call a synaptic mechanosensor that we could study both in a HEC293 line, but also importantly, the same features we saw in HEC293 were expressed in primary cortical neurons. So we've lost nothing in that transition from a cell line to a pure neuronal culture. And we scanned the physiology of the receptor, and consistent with some earlier data, showed that the change we saw, this increase in calcium influx through the receptor, is due to a change in the physiology of the receptor. Unfortunately, we don't yet know right now what mechanistically at the molecular level is occurring. So why does it lose its magnesium block? And is it physically pulling on this subunit and inducing some conformational change in that pore-forming region? We haven't carried the story forward with that. And the other thing that we realized in scanning the literature is this GluN2B subunit is highly expressed in the brain during development, as the brain is growing, as the brain is forming connections. And one of the things that we're exploring right now is when we constitutively inactivate or reduce this mechanosensing feature, does it have an impact on the development of neural circuits? And that's work that's ongoing, and we'll have an answer in about a year or so. What we then did is we transitioned to the next length scale, which is looking at circuits. And to do this, I'm going to explain a bit of the technology, a bit of the measurements that we can make, and I'll give you a sampling of some of the features, if you will, of the data that we're seeing right now. This is now under review for publication. This all started when we were scanning the literature, given that we now know that some of these sensors, some of these glutamate receptors are activated, how would it affect the circuit structure? And what we wanted to do, honestly, was to move away from some of the standard assays of measuring field potentials in a cell culture or microelectrode array technology, which gives you an inexact description of the activity of each neuron in the culture. And alternatively, we wanted to use something optically to scan for each neuron in the circuit what its activity is. So one of the properties that you see when a neuron fires an action potential is you'll get this transient calcium influx, which there are some genetically encoded calcium indicators coming out of Genelia in Virginia, which have an expression of these calcium-sensitive fluorescence markers intrinsic to neurons. So they're generating this protein all the time when you transfect the culture with a virus. And when it fires an action potential, it will show this increase in fluorescence. And relatively recently, this was a Nature Methods paper in 2009, which showed that there are new constructs available, and they've improved on these even more, that have the sensitivity to detect an increase in fluorescence with a single neuron, within a single neuron, that corresponds in their system to a single action potential firing. And as you would think, as you fire 2, 5, and 20 APs, this peak increase in fluorescence scales with that. And they showed in this paper that in their system, they can take the peak fluorescence within each neuron and extract out an activity pattern, the firing pattern, over time of each neuron in the circuit. We don't have the validation to say, for instance, a peak increase of fluorescence of 5% of baseline corresponds to 2 action potentials. What we're using in ours is a peak detection method that gives us an approximate view of the activity of the culture itself. So this is an overview of the methods, and in the next slide I'll show you what this looks like to give you a feel for some of these imaging methods. We express these genetically encoded calcium indicators, the GCAMP molecules that I'll refer to later. If you take regions of interest, these are individual neurons in the circuit, over time you can look at the increase in fluorescence for each of these neurons. This is showing 3 different neurons in the field of view, and what you can see quite readily is that there's a lot of overlap in time with these peak fluorescence patterns. You can extract, using peak detection algorithms, a map of when these are activated over time, and this is a graph where it has the different neurons in the culture identified by a distinct numeric identifier over time, and every blue spot here is associated with the firing of at least 1 action potential. So that's a peak event. But again, we don't have an ability now to say whether that's 1, 5, or even 10 action potentials. So you can look at this raster plot of activity, and you can immediately get an indication of how synchronized these neurons are. So these neurons that fall all in a line here are firing together. There's a couple of sporadic neurons that fire out of synchrony. And we have measures of defining that synchrony. And a very talented student, an MD Ph.D. student, then went into the literature and merged a couple of algorithms together to extract out not just how individuals are firing and maybe if they're in synchrony with others, but how does that develop an estimated connectivity. So this is what we call a functional connectivity. This is not a structural connectivity per se, but they're probably closely related. So what this means, for instance, if you draw a line between this green cell and this green neuron, that they're functionally connected because the pattern of activity that you extract over time, often one cell fires, that cell fires in unison with the other cell. And this is methods that in the connectomics literature are being improved every day. So this is a video of the kind of experiment that we typically get. This is a plate of cortical neurons cultured from embryonic rats. And what you'll see here is this pulsing. So there's this broad increase in fluorescence and then coming back down. The other thing is that you'll see this almost lightning storm effect where there's kind of a pock of increases in fluorescence throughout the entire field of view. This is played in real time. So this is as if you were looking through the objective of the microscope and seeing this in real time. And this is before we do anything to the circuit itself. This is leaving it alone. This is a circuit that's about 3 weeks old. So following plating, it's in the incubator for about 3 weeks. And this is the kind of activity pattern that you get. So the 2 general features. There are some times when everything fires all at once, and there are other periods where things are less synchronized. To the right, I'll show you just in a few seconds, is the same circuit now that's been mechanically injured. The effect is dramatic. And what I'll add here is, you look at this, Kazuna, if you look at this effect, this is at an hour post-stimulus. This is a level of mechanical injury that causes no cell death at 24 hours. So cells are not dying, but cells are clearly changing their activity pattern through the network. And one of the things that we're now exploring is how this whole network recovers. So again, a summary is we take raster plots, make connection maps, and what we're now studying, and I'll talk to in a minute, is how these connection maps change. And this shows you the result of the experiment I just showed you, which is pre-injury it has a certain connection map. Immediately post-injury, the only cells that are firing are these green neurons indicated here. But then at an hour, it starts to reconnect itself. There is no loss of neurons in the network. All of the neurons are intact. None of them show membrane compromise. But there's three phenotypes that I described right here. Some of them, which are in the orange squares, that they're disconnected at 5 minutes and remain disconnected. There's another phenotype where the neurons reintegrate. They may disconnect, but they come back into the network. And there's yet others which remain connected throughout. And this is intriguing because up until this point, we didn't have a tool to examine a circuit at such a scale, a single cell scale. And we're getting three generally different responses for these neurons in the population. The other thing that we can scan is what we're calling a single cell phenotype of each neuron. So each neuron expresses its relative number of postsynaptic glutamate receptors, NMDA receptors, AMPA receptors, different types of AMPA receptors. And we can use this very simple pharmacology in each neuron to block the NMDA receptor population to see how much of these calcium transients are attributed to the NMDA receptors. And then subsequently block a certain form of AMPA receptors and eventually eliminate entirely the calcium transients. But what that means for us is that we can develop a map for each neuron on the fractional contribution of each of these receptor populations. So not only do we see how the connections of each neuron changes, but we're now able to draw those changes in connectivity to its character or its phenotype. If it's dominated, for instance, by NMDA receptors, they may respond in one way relative to those neurons that are dominated by AMPA receptors. And this is a map of one neuron that we had in the circuit that I showed you at the beginning. And what you see here is in the center neuron that we're targeting, the number of connections that it makes with its neighboring neuron. This is literally over 100 different connections that we construed from the functional imaging, or the functional connectivity. And what the student did in this case was they color-coded what happened to all of these connections. How many of these connections that were formed were stable? That's in blue. How many of them were lost at any time point? This happened to be at an hour. And you can see how it's disconnected from many of the neurons that it was originally connected to. And then intriguingly, there are some neurons that it connects to that are new targets. It's a relatively small number here, but we now can look at connectivity and this sorting of connectivity to start to glean changes in the network. And this is a sample graph, and I want to leave you with two messages, this being one of them from this analysis. This is the circuit prior to a mechanical injury. Again, this injury level doesn't cause cell death. It causes a disconnection of the network, and the disconnected neurons are shown here in red. If you now go back and examine the number of connections that each of these neurons has to its neighbors, and you look at this connectivity, where the red indicates high connectivity and the blue is low connectivity, the first thing that you immediately see is that cells or neurons that are most stable have the highest number of initial connections. So it's a bit like social networks, where if you have a lot of friends, you'll be fine. If you don't have many friends, you actually are at risk. And that's what happens here when you look at a relatively low number of connections. But there's a trend here that doesn't agree with that entirely, and that is, here's the blue, where you lose many of your connections. Not many of them are stable, but there's this population here which is gaining connections. So there seems to be something else that's going on here, and in fact, going back to some of the work that I presented in the HEC293 cells, this feature goes back to the composition of the NMDA receptor. At the beginning I said the mechanosensor was associated with the gluN2B subunit. And one way of parsing out whether or not it loses connections is for each neuron, how much of the NMDA receptors have the gluN2B subunit. So those that are expressing high levels of this mechanosensitive subunit in a receptor lose a lot of connections. And intriguingly, if you don't have many of them, those are the ones that tend to form new connections. And we don't know what this means for network function at this point, because you may say that if these don't sense this initial mechanical stimulus, maybe their role is to help reestablish the network since they're not sensing the initial stimulus. We simply don't know right now. But it's bringing network structure and single cell properties back into the function of the circuit itself. So there's really three simple messages I want to convey here. The first is, as I mentioned, high connections are good. High connections in a network are actually those nodes in the network that appear to be mechanically resistant to any of the perturbations. The second is the opposite is also true. If you're not well connected from a neuron perspective in the network, you actually de-integrate from that network quite easily. And one of the features of that is you're more at risk if you have this mechanosensitive subunit. And we think that this is now giving us a different window into how these circuits are behaving. Why some of the neurons are de-integrating and others are not. And perhaps, most importantly long term, how does a network maintain its rhythmic activity after this injury? So there's some evidence in the literature that says that the activity of a circuit and the synaptic signaling that's triggered when a circuit fires on a regular basis is actually a very important trophic signal or pro-survival signal. And because the connectivity influences the rhythm of the network, and the rhythm of the network may infer survival or influence survival, we think this may give us some insight into how to recover the network function after this traumatic injury. And that's what I'm going to talk about in the last few minutes here. And that is, how do we use this to repair the circuits? So I want to call your attention to a number of papers that were published upwards of 10 years ago now, which had already identified this mechanosensing subunit, the gluN2B subunit, as something that, when it's overactivated in a fully intact receptor, it can push a neuron to a neuronal degenerative pathway. It overloads mitochondria. It's one mechanism where, if you look at the stroke literature, they tried to attack this receptor to try to improve outcome after injury. But there's this yin and yang of NMDA receptors, because just as much as you can do damage by activating certain receptor subpopulations, you can also rescue the networks by activating other populations. But none of this was really helpful to us because our neurons in these networks were not dying. They were changing their wiring, and they were changing it in a way that we really didn't know the functional implications of it. So we first went to in silico simulations where we built models at a multi-scale, at the synaptic scale, modeling the release of individual vesicles of glutamate activating these postsynaptic receptor populations. We connected these in silico networks together. We could recreate firing patterns. And we actually scanned through some of the different possibilities when you selectively lost the magnesium block of these mechanosensing NMDA receptors. And we saw one of the things that would occur immediately was an alteration in the synchrony of the network. This is the computational simulations at the spine level, the single synapse level, and how it's reflected in the raster plot predicted of the network activity and how it's synchronized at the beginning, a little bit of asynchrony, but you lose all of that organizational firing when you have this selective loss of the NMDA receptor. We went back into a culture and we proved that. We saw the exact same thing, that when you mechanically injure the circuit, you lose a lot of the synchrony in the circuit. Some of it recovers, and most importantly, if you block prior to any of these injuries the NMDA receptor, you have a very minimal change in the synchrony of the circuit. So this mechanosensing feature, again, comes into play here. And one of the ways to maintain this synchrony is to block, as I mentioned, the receptor, but also downstream If you don't block the receptor, but you block one of the consequences of this calcium influx, and this is a protease calpain, which is calcium activated, you can also help rewire or rebuild the circuits by blocking the downstream target of these mechanoactivated circuits. So that was nice, but then we went into the literature at the suggestion of another collaborator at Penn, which said, well, what's happening at your microcircuit level is this phenomenon of coherence, and how coherence in systems neuroscience, as we come to find out, is developing a widespread kind of groundswell support for the way in which circuits across different brain regions, as well as within brain regions, can synchronize information and lock in information. So this coherence, in our view, was something of the synchronous firing that we saw in the circuits. There was an investigator, the Bushaki group at now NYU Columbia, that has pioneered a lot of the approaches here. And what we then did was looked at the synchrony, or this coherence, if you will, in our circuits, and looking at ways to rebuild it. So this is a very busy slide, which simply shows you synchronized, injured, desynchronized, and importantly, when you look at this desynchronized circuit, and you didn't pre-treat with anything, one of the ways to bring back the synchrony is to block this gluon 2B subunit. This is a little surprising because you didn't think it would affect the synchronization so much. But the other important feature of this is when we started to look at models of learning and memory in the culture dish. So this is a brief slide which I'm going to show you, which talks about how you can take a circuit in culture and you can synchronize or force all the neurons to fire together. And if you do that for about 15 minutes and you remove that drug, you actually have an impression or a memory of that firing pattern in the circuitry. So what you had before and after is different, and it was because of this forced synchronization. So people have shown that this is, in a culture dish, a model of learning and memory, of long-term potentiation. And we tried to recreate this resynchronization of the circuit in injured cultures, and we could not get this potentiation. We could not get the circuit to learn after this injury. And the only way that we could recreate that learning phenomenon or phenotype is when we block these gluon 2B NMDA receptors after the stretch. And we think that that's intriguing because although there's been some discussion about the role that these gluon 2B receptors have in neuronal degeneration, no one has really started to talk about them in traumatic brain injury as a way to repair the circuitry. So the thinking, if you scale what we have in a culture dish up to animals, is that if we have an animal learn a task, and at the same time we're blocking these injured receptors, they might have an ability to learn the task better by synchronizing the circuits through blocking these receptors. So it's this neuronal synchrony that is bringing us to a point where in the acute phase following the injury, where we've identified the sensors, we can identify antagonists. And the hopeful part of the story here is that there are well-developed gluon 2B antagonists with high specificity. Some of them cross the blood-brain barrier readily. The trick, though, is that we have to consider how these circuits are synchronizing themselves and rewiring themselves in this phase. And what I didn't talk about today, but is on our mind as well, is how these changes in the sensors, so these altered receptors, the physiology is altered, are eventually replaced with other receptors. So they're removed and inserted with different receptors, and that is driving what we call a remodeling phase, which may identify different therapeutic targets. And this is, as I mentioned, in the days to weeks following the injury. So what I want to leave you with are a couple of messages from this part, which is, one, at the mechanistic scale, we're seeing this phenomenon of synchrony and its impact on, quote, learning or potentiation, and how we think drug targets may be more and more influenced by our knowledge of this synchrony, reestablishing synchrony to recover the circuits in these mild forms of traumatic brain injury. And it may indeed, in the long run, give us alternative targets. And as we look forward beyond that, I think one of the challenges for the field, and I was talking about this with some of the students at lunch, was when you look at recovery in the brain, it's very much a moving target, and concussions are no different. So we've identified changes at the receptor level, which can disappear as these receptors are replaced. The circuits are now different because they have different receptors. That's going to require some patience on our part because we have to be very careful about what drug we give when. And I think in the long run, the other aspect of the field that is now staring at us is the changes in the circuitry that occur within days to weeks. How much of that do we carry with ourselves years following the injury? Is there, for lack of a better phrase, a rewiring that we all experience, some of which may result in neurodegeneration later in life? And there are some clues that we have that we're working on right now. So with that, I'd certainly like to thank your attention. Thank you again for the invitation. I want to highlight a couple of students here. Tappan is the MD Ph.D. student that did a lot of the connectivity imaging. Pollack has now graduated from our lab and did a lot of the mechanosensor work and certainly the funding sources and some of the collaborators as well. So thank you.
Questions?
Very nice, David. I was wondering, so as I understood it, these studies were primarily done in response to a simulation of a single traumatic event. I was wondering if you had any sense or done any studies that looked at perhaps repeated lower magnitude sorts of traumas.
We have, and the protocol that we've used, whether it's right or wrong, I'm not even going to get into that. Because with repeated injuries, the biggest challenge with repetitive injuries when we talk about brain injuries is how close together should you model these injuries, and should you model low severity, high severity, low severity. There's all these different permutations. So to make it simple, we did very low severity, which is lower than all the data that I showed here, and we did it spaced a day apart. And what we're finding is that first event does precondition these circuits to be even more responsive the next time around. And one of the reasons is that there seems to be this turnover at the NMDA receptor level, which inserts more of these mechanosensitive receptors after the first stimulus. So it's almost like nature is pushing us down a more sensitive path. And ultimately, over time, those will be replaced more and more by mechanically insensitive receptors, but there's a vulnerability there. So that's what we're seeing. In a culture dish, we can eventually look at animals as a platform.
So you mentioned that the cell culture that you use are from embryonic rats, and you culture them for 3 weeks?
Yeah.
So how do you think age plays into all of this? Because, I mean, from what you have, it's a 2D culture that's plated. So let's say, you have a 3-week-old rat versus, 6-month-old rat. I mean, how do you think neuroplasticity changes?
Yeah. One of the things that we know is of the different subtypes of NMDA receptors, the one that appears first in development are these NR1, NR2Bs. But over time, and if you look at humans as well as in rats, in the fully mature state, you have NR1, NR2A, NR2B triheteromer receptors. So what you have is this intermediate sensitivity. In most of our 3-week-old cultures, when we characterize how much of the young receptors versus the mature receptors, it turns out it's about 1/3 young, 1/3 of these triheteromer, and there's a 1/3 in between. So it's not fully adult. It's on its way to being adult-like, but it's not yet there. And I think it influences it quite a bit. So this is one of those, I wouldn't say it's a secret, but one of the things that we've realized in our group is when you read a paper that uses a primary cortical neuron culture, you have to really look into the methods, because we found that even a difference of 4 or 5 days makes a big difference in the response. The challenge that we have is it's hard for these cultures to stay alive more than about 3 weeks on the surfaces that we're treating. So we're a little bit limited in how far out we can go.
And then most of the raster plots and the responses that you've shown are, well, with respect to the antagonist, we're on a 10-second time scale,
Yeah.
right? The asynchrony starts 10 seconds after.
Yeah.
And I guess I was wondering, when you put in the antagonist, are you putting it, you're putting it right at the time, right after the stretch happens, right? Or
so we actually, it's about 30 seconds after.
okay.
And there's a reason for that. I won't get into it. It's a technical detail.
Have you tried later applications, or like days after, or?
We haven't. It's a good idea. We see that this persistence in asynchrony will occur at a day and 3 days after the injury, but we don't have the data to show we block at that point. You
If there's
can
any
break that.
Gotcha.
It's a good idea.
So I'm trying to understand what actually is going on mechanically around the regional receptor when you apply your stretch. You have like more, so it's not, if I understand correctly, it's not the membrane tension that is the signal, right? It's more the relative distance between the membrane and the cytoskeleton.
Yeah, and so we know in other experiments that if you depolymerize the actin, you completely eliminate the response. So the actin structure is important. We've also tried to dope the membrane to increase the fluidity, decrease the fluidity. That seems to have a much less significant impact on the response that we see. So if it was cytoskeleton versus membrane, we think it's primarily cytoskeleton. And we think it is primarily through the actin linkages and actin-associated proteins, because they are binding sites for alpha-actin, for instance. And the other thing, and I didn't have a chance to talk about it, these are dynamic interactions that occur between these actin-associated proteins and the subunit, the C-terminus. And when you take this recombinant receptor model and you stretch it to the same level, but you do it very slowly, you get no response. So there's a rate sensitivity to it, which I think has to do with this dynamic interaction with the cytoskeleton and the subunits. So that's why, if you talk to neurosurgeons, if they insert a deep brain stimulator probe, they go very slowly because they've realized as young residents, when they're impatient and they go quickly, they can do some damage. And so this rate sensitivity has interesting features to it. So if you press on the brain of an animal very slowly, the cortex, you really don't create much of a functional change that you can see in the animal. But obviously, if you do the same pressing very quickly, you can create an injury.
Swell neurons?
Pardon me?
Can you actually swell them?
Yeah, you can. We've done it. This is what happens when you swell them is they release neurotransmitter at a higher frequency. So you kind of alter the dynamics of it. It gets complicated. So you can add sucrose, you can do all these little tricks. And they actually have a very narrow operating range in terms of osmolarity. If you go too high or too low, the neurons just check out and they die pretty quickly.
Let us take one final question. I think you had a question back there. No?
I did, but it was...
Okay. And I saw a hand go up here.
Well, I
Yeah.
just had so one of the messages you mentioned before is that it seems like the neurons that have more connections are more resistant to injury. I'm not very familiar with neurobiology. I was just curious about what determines how many connections a neuron has and whether that can be changed.
I don't think anybody knows. As far as I can see in the literature, they're just beginning to, even in a culture dish, map out the distribution of the network that forms. And this is where there's a lot of activity in network science nowadays, some of which applies to neurobiology. But to my knowledge, no one knows why one is highly connected, or let's say double the connectivity that its neighbor. So.
With your permission, I'm going to bring this to an end. There's a taxi waiting for you out there. But before we go, you said you like the team prototyping facility. So we said, how about if we prototype something for him using the team prototyping facility? So we have designed and made this memento here that says Biomedical Engineering, UC Davis, Department of Biomedical Engineering, University of California, Davis, recognizes David Meaney for outstanding contributions in the field of biomedical engineering, Distinguished Speaker Series 2012-2013. And here it says that it is designed and fabricated in the team prototyping facility. So thank you very much for taking the time to come over here. We enjoyed this presentation.